Okay, so in order to understand how to key out seaweeds, we have to talk a little bit about morphology. It's quite like a plant, like a land plant, but it's very, very different in the way that these organisms function. So while land plants have specialized cells for different functions, like transferring nutrients and water across the body of the plant, seaweeds, the cells are generally not specialized, but kind of they all have potential to do photosynthesis, potentially reproduce, and so on. So, but the structures look similar. They can be analogous. So with kelps or any of the seaweeds, we have what we call a holdfast. This can be kind of claw-like. It can be discoid, like you see here. And it's a good characteristic to look at for a lot of species when you're trying to identify them from closely related or very similar looking ones. We have what we call a stipe, which can then turn into a midrib within a blade. That's very similar to like a stem or a trunk. And then we have a frond or a blade, which is quite like a leaf. Generally, we'll say blade, and this can be around a midrib. It can be reticulated like you see here. This is basically sugar kelp right here. There can be air bladders or reproductive structures within that blade. So here's an example of a species. This would be like Dictyota here with reproductive structures throughout the blade. And then it can have very different branching patterns, which you might then say, you might start calling them branches or fronds. Some of the branching patterns that we might see that are very useful for helping key out, especially the red algae. You have alternate branching, so every other branch is going a different direction. You have pinnate or disticus branching, quite like a feather. Unilateral or secund, so branching only on one side. There can be spiral branching, so it moves around the central axis or this midrib, quite like maybe like a staircase. You have whorl branching, so branching at one location in multiple directions. Decussate, meaning it's almost like alternate branching, but on two sides. We have dichotomous branching, so forked branching in a series, trichotomous branching in three directions in a series, and then cervicorn, which is quite pretty, so you'll see this in coxcomb in a few species that we'll talk about, where you have kind of U-shaped branching, for lack of a better word right now, in a unilateral plane. We also have a lot of crustose morphologies. So if we look here, here's some examples of coralline algae. These are from tropical reefs, but they function the same way on a temperate reef. I actually think that the one here on the right is from a temperate reef, and maybe the source is marlin. Basically, encrusting forms can mimic what is beneath them. So you can see that here with this coral bunny. They can kind of have their own fan-shaped structure, which is what you see here with this Tritanoderma species. They can also be free-living crustose morphs. So that's generally we call that a rhodolith. They form free-living balls that then roll around, and they can form their own habitats, rhodolith beds that are very productive, mostly in secondary production, so a habitat for other organisms. And they can also form what we call marl beds, although this is generally referred to as a branching coralline algae.